Let's know about lipids. Lipids are of great importance to the body as the cheap concentrated storage form of energy. Besides their role in cellular structure and various other biochemical functions, such as lipids are a heterogeneous group of compounds. Lipids may be regarded as organic substances relatively insoluble in water, soluble in organic solvents, alcohol, ether, etc. actually or potentially related to fatty acids and utilized by the living cells. Unlike the polysaccharides, proteins, and nucleic acids, lipids are not polymers. They are mostly small molecules. Now let's know about classification of lipids. Lipids are broadly classified, modified from blood into simple, complex, derived, and miscellaneous lipids, which are further subdivided. Now let's know about simple lipids. Esters of fatty acids with alcohols. These are of mainly are of two types: fats and oils. These are esters of fatty acids with glycerol. The difference between a fat and oil is only physical. Thus, oil is a liquid while fat is a solid at room temperature. Now, let's know about waxes. Esters of fatty acids with alcohols other than glycerol. Cetyl alcohol is most commonly found in waxes. Esters. Now let's know about the classification of lipids. Lipids are broadly classified into simple, complex, derived and miscellaneous lipids, which are further subdivided. Now let's know about simple lipids. Esters of fatty acids with alcohols. These are mainly are of two types: fats and oils. These are esters of fatty acids with glycerol. The difference between fat and oil is only physical. Thus, oil is a liquid while fat is a solid at room temperature. Now, let's know about waxes. Esters of fatty acids, usually long chain with alcohols other than glycerol. Cetyl alcohol is most commonly found in waxes. Now, complex lipids. Esters of fatty acids with alcohols containing additional groups such as phosphate, nitrogenous base, carbohydrate, protein, etc. They are further subdivided into phospholipids, glycolipids, lipids, and other complex lipids. Let's know about phospholipids. Lipids containing phosphoric acid and frequently a nitrogenous base. This is an addition to alcohol and fatty acids. Now let's know about glycolipids. These lipids contain a fatty acid, carbohydrate and nitrogenous bases. The alcohol is sphingosine, hence they are also called as glycosphingolipids. Glycerol and phosphate are absent. Example: cerebrosides, gangliosides. Next point is lipoproteins. Macromolecular complexes of lipids with proteins. Other complex lipids: sulfolipids, aminolipids, and lipopolysaccharides are among the other complex lipids. Third point is derived lipids. These are the derivatives obtained on the hydrolysis of group 1 and group 2 lipids, which possess the characteristics of lipids. These include glycerol and other alcohols, fatty acids, mono and diacyl glycerols, lipid soluble vitamins, steroid hormones, hydrocarbons, and ketone bodies. 4 is miscellaneous lipids. These include a large number of compounds possessing the characteristics of lipids, example carotenoids, squalene, hydrocarbons such as pentacosane or terpenes in beeswax, that is pentacosane. Neutral lipids. Let's know about neutral lipids. The lipids which are uncharged are referred to as neutral lipids. These are mono, di, and triglycerols and cholesterol esters. Now let's know about functions of lipids. Lipids perform several important functions. First point is they are the concentrated fuel reserve of the body. that is triacylglycerols. Second is lipids are the constituents of membrane structure and regulate the membrane permeability. Phospholipids and cholesterol. They serve as a source of fat soluble vitamins, that is A, D, E, K. Lipids are important as cellular metabolic regulators, steroid hormones and prostaglandins. Now let's know about fatty acids. Fatty acids are carboxylic acids with hydrocarbon side chain. They are simplest form of lipids. Even and odd carbon fatty acids. Let's know about even and odd carbon fatty acids. Most of the fatty acids that occur in natural lipids are of even carbon, usually 14C to 20C. This is due to the fact that biosynthesis of fatty acids mainly occurs with the sequential addition of two carbon units. Palmitic acid and stearic acid are the most common. Among the odd chain fatty acids, propionic acid and valeric acid are well known. Now let's know about saturated and unsaturated fatty acids. Saturated fatty acids do not contain double bond, while unsaturated fatty acids contain one or more double bonds. Both saturated and unsaturated fatty acids almost equally occur in the natural lipids. Fatty acids with one double bond are known as monounsaturated, and those with two or more double bonds are collectively known as polyunsaturated fatty acids, PUFA. Now let's know about shorthand representation of fatty acids. Instead of writing the full structures, biochemists employ shorthand notations to represent fatty acids. The general rule is that the total number of carbon atoms is written first, followed by the number of double bonds, and finally the position of double bonds starting from the carboxyl end. Thus, saturated fatty acid, palmitic acid is written as 16 is to 0, oleic acid is 18 is to 1 is to 9, arachidonic acid as 20 is to 40, 5, 8, 11 and 14. Now let's know about essential fatty acids. The fatty acids that cannot be synthesized by the body and therefore should be supplied in the diet are known as essential fatty acids. Chemically, they are polyunsaturated fatty acids, namely linoleic acid and linolenic acid. Arachidonic acid becomes essential if its precursor linoleic acid is not provided in the diet in sufficient amounts. Now let's know about triacylglycerols. Triacylglycerols are the esters of glycerol with fatty acids. The fats and oils that are widely distributed in both plants and animals are chemically triacylglycerols. They are insoluble in water and non-polar in character and commonly known as neutral fats. Now let's know about fats as stored fuel. Triacylglycerols are the most abundant group of lipids that primarily function as fuel reserves of animals. The fat reserve of normal humans is sufficient to meet the body caloric requirement for 2 to 3 months. Now let's know about structures of acylglycerols. Monoacylglycerols, diacylglycerols and triacylglycerols respectively consist of 1, 2 and 3 molecules of fatty acids esterified to a molecule of glycerol are known. Among these, triacylglycerols are the most important biochemically, and triacylglycerols of plants have higher content of unsaturated fatty acids compared to that of animals. Now let's know about phospholipids. These are complex and or compound lipids containing phosphoric acid in addition to fatty acids, nitrogenous bases, and alcohol. There are two classes of phospholipids. First point is glycerophospholipids, and that contain glycerol as the alcohol, example lecithins. cephalins, phosphatidylinosytol, phosphatidylserine, and plasmalogens. Second is sphingosphospholipids that contain sphingosine as the alcohol. Ceramide. Example, ceramide. Now let's know about lipoproteins. Lipoproteins are molecular complexes of lipids with proteins. They are the transport vehicles for lipids in the circulation. There are five types of lipoproteins, namely chylomicrons, very low density lipoprotein, that is in short VLDL, low density lipoprotein LDL, high density lipoprotein HDL, and free fatty acid albumin complexes. Now let's know about steroids. Steroids are the compounds containing a cyclic steroid nucleus named that is namely cyclopentano perhydrophenanthrin, in short CPP. It consists of a phenanthrin nucleus ring, that is ABC, to which a cyclopentene ring is attached. There are several steroids in the biological system. These include cholesterol, bile acids, vitamin D, sex hormone, and adrenocortical hormones. If the steroid contains one or more hydroxyl groups, it is commonly known as sterol. The structures of steroid nucleus cholesterol we will discuss now. Now let's know about proteins and amino acids. Proteins are the most abundant organic molecules of the living system. They occur in every part of the cell and constitute about 50% of the cellular dry weight. Proteins form the fundamental basis of structure and function of life. Now let's know about function of proteins. Proteins perform a great variety of specialized and essential functions of the living cells. These functions may be broadly grouped as static and dynamic. Structural functions. Certain proteins perform brick and mortar roles and are primarily responsible for structure and strength of body. These include collagen and elastin found in bone matrix, vascular system and other organs, and alpha keratin present in epidermal tissues. Now let's know about dynamic functions. Let's know about functions of proteins. Proteins perform great variety of specialized and essential functions in the living cells. These functions may be broadly grouped into static and dynamic. Structural functions. Certain proteins perform brick and mortar roles and are primarily responsible for structure and strength of body. These include collagen and elastin found in bone matrix, vascular system and other organs, and alpha keratin present in epidermal tissues. Now let's know about dynamic functions. The dynamic functions of proteins are more diversified in nature. These include proteins acting as enzymes, hormones, blood clotting factors, immunoglobulins, membrane receptors, storage proteins. Besides their function in genetic control, muscle contraction, respiration, etc., proteins performing dynamic functions are appropriately regarded as the working horses of cell. Now let's know about proteins are polymers of amino acids. Proteins on complete hydrolysis with concentrated HCl for several hours yield L alpha amino acids. This is a common property of all the proteins. Therefore, proteins are the polymers of L alpha amino acids. Now let's know about amino acids. Amino acids are a group of organic compounds containing two functional groups, amino and carboxyl. The amino group is basic while the carboxyl group is acidic in nature. Now let's know about general structure of amino acids. The amino acids are termed as alpha amino acids if both the carboxyl and amino groups are attached to the same carbon atom. The alpha carbon atom binds to a side chain represented by R which is different for each of the 20 amino acids found in proteins. The amino acids mostly exist in the ionized form in the biological system. Now let's know about optical isomers of amino acids. If a carbon atom is attached to four different groups, it is asymmetric and therefore exhibits optical isomerism. The amino acids possess four distinct groups, that is R, H, COO minus NH3 plus, held by alpha carbon. Thus all the amino acids have optical isomers. The structures of L and D amino acids are written based on the configuration of L and D glyceraldehyde. The proteins are composed of L alpha amino acids. Now let's know about classification of amino acids. There are different ways of classifying the amino acids based on the structure and chemical nature, nutritional requirement, metabolic fate, etc. Now let's know about amino acid classification based on the structure. A comprehensive classification of amino acids is based on their structure and chemical nature. Each amino acid is assigned a three-letter or one-letter symbol. These symbols are commonly used to represent the amino acids. The 20 amino acids found in proteins are divided into 7 distinct groups. Now let's know about nutritional classification of amino acids. The 20 amino acids are required for the synthesis of variety of proteins besides other biological functions. However, all these 20 amino acids need not be taken in the diet. Based on the nutritional requirements, amino acids are grouped into two classes, essential and non-essential. Essential or indispensable amino acids. Let's know about essential or indispensable amino acids. The amino acids which cannot be synthesized by the human body and therefore need to be supplied through the diet are called essential amino acids. They are required for proper growth and maintenance of the individual. The 10 amino acids we will now discuss. First is arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan. Now let's know about non-essential or dispensable amino acids. The body can synthesize about 10 amino acids to meet the biological needs. Hence, they need not be consumed in the diet. These are glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, tyrosine, and proline. Now let's know about structure of proteins. Proteins are the polymers of L alpha amino acids. The structure of proteins is rather complex, which can be divided into four levels of organization. First is primary structure, the linear sequence of amino acids forming the backbone of proteins. Second is secondary structure, the special arrangement of protein by wasting of the polypeptide chain. Third is tertiary structure, the three-dimensional structure of a functional protein. Fourth is quaternary structure. Some of the proteins are composed of two or more polypeptide chains referred to as subunits. The special arrangement of these subunits is known as quaternary structure. Now let's know about primary structure of protein. Each protein has a unique sequence of amino acids which is determined by the genes contained in DNA. The primary structure of a protein is largely responsible for its function. Peptide bond. Let's know about peptide bond. The amino acids are held together in a protein by covalent peptide bonds or linkages. These bonds are rather strong and serve as the cementing material between the individual amino acids. Now let's know about formation of a peptide bond. When the amino group of a amino acid combines with the carboxyl group of another amino acid, a peptide bond is formed. Note that a dipeptide will have two amino acids and one peptide bond. Peptides containing more than 10 amino acids are referred to as polypeptides. Now let's know about determination of primary structure. The primary structure comprises the identification of constituent amino acids with regard to their quality, quantity and sequence in a protein structure. A pure sample of a protein or a polypeptide is essential for the determination of primary structure, which involves three stages. First is determination of amino acid composition. Second is degradation of protein or polypeptide into smaller fragments. Third is determination of the amino acid sequence. Now let's know about secondary structure of protein. The conformation of polypeptide chain by twisting or folding is referred to as secondary structure. The amino acids are located close to each other in their sequence. Two types of secondary structure, that is alpha helix and beta sheet, are mainly identified. Now let's know about alpha helix. Alpha helix is the most common spiral structure of protein. It is rigid arrangement of polypeptide chain. Alpha helical structure was proposed by Pauling and Corey in 1951, which is regarded as one of the milestones in the biochemistry research. The silent features of a right-handed alpha helix, which is a stable and more commonly found structure in the living system. The alpha helix is a tightly packed coiled structure with amino acid side chains extended outward from the central axis. Second is the alpha helix is stabilized by extensive hydrogen bonding. It is formed between hydrogen atom attached to peptide nitrogen and oxygen atom attached to peptide C. Third point is all the peptide bonds except the first and last in a polypeptide chain participate in hydrogen bonding. Fourth is each turn of alpha helix contains 3.6 amino acids and travels a distance of 0.54 nanometer. The space of each amino acid is 0.15 nanometer. The alpha helix is a stable conformation formed spontaneously with the lowest energy. Beta plated sheet. This is the second type of structure proposed of Pauling and Corey. Beta plated Beta plated sheets are composed of two or more segments of fully extended peptide chains. In the beta sheets, the hydrogen bonds are formed between the neighboring segments of polypeptide chains. Now let's know about tertiary structure of protein. The third dimensional arrangement of protein structure is referred to as tertiary structure. It is a compact structure with hydrophobic side chains held interior while the hydrophilic groups are on the surface of the protein molecules. This type of arrangement ensures stability of the molecule. Now let's know about bonds of tertiary structures. Besides the hydrogen bonds, disulfide bonds, ionic interactions and hydrophobic interactions also contribute to the tertiary structure of proteins. Now let's know about domains. The term domains is usually used to represent the basic units of protein structure and function. The polypeptide with 200 amino acids normally consists of two or more domains. Now let's know about quaternary structure of proteins. A great majority of the proteins are composed of a single polypeptide chain. Some of the proteins, however, consist of two or more polypeptides which may be identical or unrelated. Such proteins are termed as oligomers and possess quaternary structure. The individual polypeptide chains are known as monomers, protomers, and subunits. That a dimer consists of two polypeptides while a tetramer has four bonds in quaternary structure. The monomeric subunits are held together by non-covalent bonds, namely hydrogen bonds, hydrophobic interactions, and non and the ionic bonds. Now let's know about classification of proteins. Proteins are classified in several ways. Three major types of classifying proteins based on their function, chemical nature and solubility properties and nutritional important we will discuss now. Now let's know about functional classifications of proteins. Based on the function they perform, proteins are classified into different groups. Structural proteins, that means keratin of hair and nail collagen of bone. Second is enzymes or catalytic proteins, hexokinase, pepsin. Third is transport proteins, example hemoglobin, serum albumin. Fourth is hormonal proteins, that is example insulin, growth hormone. Fifth is contractile proteins, actins and myosin. Storage protein, that is ovalbumin, glutein. Seventh is genetic proteins, nucleoproteins. Eighth is defense proteins, that is snake venom, immunoglobulins. Nine is receptor protein for hormones, viruses. Now let's know about protein classification based on the chemical nature and solubility. There is a more comprehensive and popular classification of proteins. It is based on the amino acid composition, structure, shape and solubility properties. Proteins are broadly classified into three major groups. One is simple proteins, second is conjugated proteins, and third is derived proteins. Let's know about simple proteins. They are composed of only amino acid residues. Second is conjugated proteins. Besides the amino acids, these proteins contain a non-protein moiety known as prosthetic group or conjugative groups. Third is derived proteins. These are the denatured or degraded proteins, products of simple and conjugated proteins. These three classes of proteins are further subdivided into different groups. Among the small, among the simple proteins, globular proteins are spherical in shape, soluble in water or other solvents and undigestible. Example: albumin, globulin. Scleroproteins are fiber-like in shape, insoluble in water and resistant to digestion. Example: collagen, keratin. The conjugated proteins may contain prosthetic groups such as nucleic acid, carbohydrate, lipid, metal, etc. The primary derived proteins are produced by agents such as heat, acids, alkalis, etc., while the secondary derived proteins are hydrolytic products of proteins. Now let's know about isoprenoids and pigments. Isoprenoids and pigments are organic compounds mostly distributed in plant kingdom. They perform a wide variety of functions. Now let's know about isoprenoids. Isoprenoids are also called as terpenoids or as they are found in turpentine oil in high concentrations. The naturally occurring isoprenoids are composed of a 5 carbon isoprene units. A majority of the isoprenoids are formed by joining of isoprene units head to tail. Now let's know about classification of turpentine. The classification of terpenes is mainly based on the number of isoprene units present. The major classes of terpenes is selected. Examples we will discuss. First is pigments. Pigments are colored organic compounds found in living organisms, mostly in plants and to a minor extent in animals. Chemically, pigments are high molecular weight molecules, mostly composed of unsaturated hydrocarbons. Some of the pigments also contain cyclic structures. The major group of pigments now we will discuss. First is tetrapyrols. Let's know about tetrapyrols. The most abundant colored compound in the world is chlorophyll. The photosynthetic pigment. There are different types of chlorophyll C, D, E, A with sunlight variation in colors green, greenish blue, greenish yellow. Structurally, chlorophylls are composed of tetrapyrols, that means pyrrole rings is available with their nitrogen linked to magnesium. Tetrapyrols are also found in heme in certain proteins. These include hemoglobin, cytochromes, catalase and peroxidase. Now let's know about tetraterpenes or carotenoids. The color of carotenoids is variable, generally yellow, orange or red. A large number of carotenoids have been identified in plant kingdom. Example: beta carotene, xanthophylls, lycopene. Now let's know about anthocyanins. Anthocyanins are a group of flavonoids which represent the natural phenolic acid products. Anthocyanins are colored compounds mostly found in flowers and fruits. They contain a common ring structure called anthocyanidin. Now let's know about quinoid pigments. Being present in trace amounts, quinoid pigments do not significantly contribute to visible colors. They, however, perform some other functions, example, involvement in electron transport chain, antioxidant functions, etc. The most common quinoid pigments are benzoquinones, naphthoquinones, anthraquinones, and tannins and lignins. See, from proteins, this is a protein, simple protein. This is a total structure, protein structure. This is simple protein. Proteins is divided into three parts. One is simple protein, and there is conjugated, and next is derived. All the classifications I have written. You can write these classifications to remember early. Finally finished. Any questions regarding this, you can ask me in the comment box, and for more updates you can subscribe my channel. Thank you.